Hi everyone, this is the simulated epidemic lab. So in this lab, we were going to simulate what would happen if we have an outbreak of disease. And we were going to talk about the significance of that. But seeing with everything that's going on with coronavirus, I think everyone in class understands the significance of how bad an outbreak can be. So we're going to discuss some background about epidemics and then what the experiment would have been if we were actually doing it in lab. Okay, to give some background, epidemiologists, here, let me move myself. Okay, epidemiology is the field of science that studies the location, timing, and transmission of disease. So if you were an epidemiologist, like if we think about the coronavirus situation and what's going on, what epidemiologists did is they saw that there was a disease outbreak. So they look at how it's being transmitted. Is it direct contact? Is it airborne? Is it fecal oral? Is it like some contaminated food that's passing it? So with the transmission, we epidemiologists taught us that with coronavirus, the outbreak, the transmission is person to person and it's through direct contact. It's through droplets. They study location of disease because understanding the location and the outbreak is very important. So with coronavirus, the beginning of the outbreak started in China, in Wuhan, China, and the timing of disease is also important. Is a certain outbreak or infection seasonal? Does it only spread in the summer or winter? So this is all important for epidemiologists to gather this data to come up with a good way for all scientists to tackle an outbreak together. So that's epidemiologists and microbiologists and scientists and medical doctors and all the healthcare workers take all of this information so that we can better understand the disease with the goal of controlling or preventing the spread of disease. Because once we have an epidemic, it can quickly turn into a pandemic, as we've all seen with coronavirus. So what you gather all this data in epidemiology and you try to come up with some conclusive way to control the disease or prevent it. Excuse me, right now, you guys all know with the coronavirus outbreak pandemic, the controlling method is just having everyone wear masks, quarantining, and we'll learn more about that in lecture. So epidemiologists track the occurrence, the spread of a disease, and they gather case numbers, who's sick, how many people are sick, what country, where is it spreading? Is it spreading in South Korea or Italy or the US or different countries more? And we're going to talk about different terms with disease outbreak. So these are endemic, epidemic, and pandemic. Endemic is the expected low level of a disease. So if you have maybe like a country or a classroom and you have a few, maybe like 2 or 3 people that have a coronavirus, that would be an endemic level. It's just the expected low number of disease. Epidemic is when the incidence goes much higher in a population than expected. Pandemic is when a disease becomes global and it's affecting people on several continents, several countries. So with coronavirus, it started out epidemic. Well, it started out actually endemically, started out a few cases in Wuhan, that spread, it became an epidemic, and now it's in every single country it's been detected, so that's pandemic. And pandemic comes from Pangea, and this is where all the continents were one before they spread out. So that's where the term comes from. So endemic is expected low levels of a disease. Epidemic is when it's greater than expected levels, and pandemic is when we have a disease spread globally in multiple countries. And epidemiologists always want to find the source of disease outbreaks. The person, the animal, the water source, the event source, all of this is very important for them to gather. And a very important term I want everyone to know is index case. Index case is the first person to have the disease which has led to an outbreak. It's very important to determine the index case because it can tell you a lot about the spread of disease. Where did it Is the index case, I know this has happened before with the spread of disease, is it someone in prison who ate lettuce from a farm nearby? So all of these things are important, and the farm and lettuce example is a real example. Once we had an E. coli, a certain strain of E. coli that was causing diarrhea, so it was at epidemic levels, and that was the story, is that there was lettuce in a farm that had been contaminated, and that lettuce was sent to a prison, and people in the prison were eating the lettuce. So it's important to understand the index case. Who's the first person to have the disease, and how are they moving? And so in this lab, you were supposed to simulate the spread of an epidemic through direct contact handshaking. So our disease in lab, the way it spread, its transmission is through direct contact. It's through handshaking. And we can think about this similar with coronavirus. If someone is infected and they sneeze on their hand and then you shake hands with them and you touch your face, you can get coronavirus. And so we were trying to see how fast a disease can quickly spread, and we were going to gather all the data and determine who was the first person infected. The organism we were using in the outbreak, so our like coronavirus, was Micrococcus luteus, which is a bacterium. And the reason we were using this bacteria is because it grows distinct yellow colonies on agar plates. This is what Micrococcus luteus looks like. So we can determine what happened with the outbreak. And actually, before I get to the objectives, I want to talk about a little about how the experiment was supposed to be done. So what was supposed to happen is everyone was supposed to get a piece of candy that you're not supposed to eat. And one of the pieces of candy has Micrococcus luteus on it. We don't know which student that is because they all look the same. And what you guys were supposed to do is you're supposed to wear gloves, and you're supposed to take the piece of candy, which everyone has a regular piece of candy. One person has a piece of candy that has Micrococcus luteus, and you're supposed to spread it all around everywhere on your hands. And then we were supposed to go around shaking hands just to simplify the experiment. And then after shaking hands, we would take an agar plate, and you would take your fingers and you would put it on the agar plate so that we could determine who was infected. And we could easily see Micrococcus luteus because it forms these nice yellow colonies. And the conclusion of this lab, if you guys had been doing it, is we do round one of handshaking and then we do round two. And by round two, almost everyone is infected. So this is why this agar plate is split into two areas. And it was supposed to teach you that disease can spread really quickly, which you guys have all learned with this unfortunate real life example with the COVID-19. So the objectives of this lab is for you guys to define epidemiology, epidemic, and pandemic. So know what these terms mean, define index case, given a plate, identify the simulated epidemic experiment, what was the transmission mode for this simulated epidemic, and identify the bacteria used in this experiment and explain why. So with epidemiology, remember epidemiologists study three main things: location, timing, and transmission of disease, and their goal is to control and prevent an outbreak. An epidemic is when a disease is greater than normal levels or expected levels. Pandemic is when the disease spreads all throughout the world, different countries, multiple countries. Index case is the first person infected with the disease, and epidemiologists try to determine who the index case is. Given a plate, identify the simulated epidemic experiment. This is what the experiment plates would have looked like. And we had Micrococcus luteus. We used Micrococcus luteus because they form very distinct yellow colonies. So in case someone had some other bacteria on their gloves, we can differentiate the infection versus just a random other bacterial colony. And the transmission mode for this simulated epidemic is we were doing direct contact handshaking. And we saw that our outbreak that started off with one person in class having an infected piece of candy spread in the whole class. And this is a very realistic example of what's going on in the world today. So I hope that at least from this lab, you guys just leave knowing what these terms are, knowing what epidemiology is: epidemic, pandemic, index case, transmission mode. And it's a pretty simple, fun lab. Unfortunately, we're not doing it.